Have you ever asked yourself the question, what's age? What's youth? We recognize it instantly. The way they look, the way they move, the way they don't hesitate. And then there comes a time where you do. You hesitate before you run, before you lift, before you trust your body. Something has changed. I tried to find a definition, and what I found was surprisingly vague. Epidemiologists define youth as an age range. Biologists define it as the period before maturity, before we age. In other words, it's defined by what it's not, and that's a weak definition. I'm a neurosurgeon at the University of Cambridge with a specialist interest in complex surgery of the spine. The condition I treat most frequently is called Degenerative Cervical Myelopathy. You've probably never heard of it, but you should. It affects between 1% and 2% of adults. It's caused by wear and tear changes of the neck, compressing the spinal cord. At the beginning, symptoms are often subtle. Some tingling in your fingertips or numbness, clumsy hands, imbalance, bladder urgency. But over time, I've seen patients go from being able to hold a pen and writing to not being able to hold a fork. Sometimes it causes paralysis from the neck down. So I often refer to it as a slow-motion spinal cord injury. The greatest risk factor is age. In other words, in my work, youth determines whether function gets lost and whether it returns. But it never quite does, like in so many other age-related conditions. So it's no surprise that aging has become an intense focus of research over the last decades. The standard model of aging is that cells accumulate damage over time caused by the universal force of entropy. The hallmarks of aging describe what goes wrong inside of the cells and the tissues. Mitochondria slow down, epigenetics gets deranged, the whole system becomes fragile. And aging can be seen as a sum total of the hallmarks. Here's the issue. Cells aren't static. They're dynamic systems. Molecules turn over. And that leads to a paradox. If everything's constantly replaced, why doesn't the damage simply go away? Why doesn't it wash out? And maybe we've been asking the wrong question. Instead of asking what's age, maybe we should focus on the question, what's youth? What if youth isn't the absence of damage, but the presence of repair? Damage happens constantly, to all of us, all the time. The difference is, in youth, repair is faster than damage. Aging begins when repair can't keep up. It's not a clock, it's a rate problem. During my training, I realized that surgery can fix anatomy, but it can't restore biology. And so I went back to the lab to study stem cells. And these are extraordinary cells, the origin of all the cells in our body. And they're also unique because they actively stay young. They have to. If they aged, our children could inherit age, and most likely our species wouldn't last. So we ask a simple question: how do stem cells remain young? And in order to answer that, we wanted to make them old. If youth is a state of heightened repair, we should be able to overwhelm it. Nothing in biology is absolute. So we tried many things. We irradiated them. We treated them with chemicals that damage the mitochondria. But whatever we tried, we got a subset of these aging hallmarks, not the entire picture, until we came across a remarkable condition, Hutchinson-Gilford progeria, a rapid aging disease. It's caused by a single mutation that produces a protein that's called progerin. And progerin affects the cell nucleus and causes accelerated aging. By the way, exactly that protein, progerin, also accumulates in the natural aging skin. So when we introduced the progerin into the stem cells, they became truly old. They displayed all the hallmarks of aging. We had created something that shouldn't exist. Cells designed to remain young, becoming old. And then we asked, what happens when we stop it? Would they stay old, like any other cell type once exposed to progerin? Within 2 weeks, the stem cells repaired, and they reversed all the aging hallmarks. They became young again. And this gave us a powerful system to study aging and rejuvenation. We were able to conduct large-scale experiments, and after 3 years of systematically testing every single gene in the human genome, more than 20,000, we used CRISPR technology to do that. We found 151 genes that were regulating aging and rejuvenation across all the cells and tissues that we looked at. Here's the key insight. There's not a single master switch. Each of the genes controls specific repair programs for specific aging hallmarks. Aging is not a single thing, and neither is rejuvenation. But of course, stem cells aren't patients, and aged stem cells, very artificial. So we turned to skin cells from donors of different ages. And to test these more efficiently, we created an AI system that we called IMAGE that can estimate the age of a cell from microscopic images. IMAGE correlates very well with chronological age. What we found was striking. The same repair matters matter. And when we modulate these programs, we can partially restore youth. We then looked at existing FDA-approved drugs. 15% of them were able to regulate the age of a cell. 5% showed strong age reversal, like the one in the image here. This means there are drugs and there are also supplements out there that can already influence aging biology. So rejuvenation may not be magic. It may just be modulation of repair. And there's not a single solution because there isn't a single cause. True rejuvenation requires a combinatorial approach. But if you find the right combination, then the impact can be quite profound. Mice that are 2 years old show distinct signs of aging. But if treated with the right cocktail, these can be reversed, not only on the surface, when you look at the coat, also in terms of function. And while old mice struggle to hold on to the top of the cage, those treated regain strength and activity. So what have we learned? We don't age because damage happens. Damage has always happened. We age because repair slows down. And what troubles me is not that we can't yet apply this knowledge, it's that we just only started to ask the right question. Applying this to patients isn't simple. DCM is complex, trials are hard, costs are enormous, especially if you want to test combinations. And while we are already at this stage conducting the first trial with a single drug, is there a way how we can speed up the testing of combinations? We may need to begin somewhere more accessible, the skin. Cosmetics may seem less important, less urgent, but sometimes the fastest path to the hardest diseases is not a straight line. You build the engine where proof comes fastest, and then you bring it where the need is greatest. DCM, Alzheimer's, Parkinson's. So let me return to the question, what's youth? Youth isn't time, it's repair. And while aging might be our fate, it's also biology, and biology can be understood. To restore youth, we don't need to defeat aging. We just need to remember how to repair. Thank you.